All right, welcome to the session on in vitro synthetic biology and molecular programming. So the main goal of this session is to take a little look at engineering systems, that are perhaps lifelike, and whether we can build them from scratch rather than building on the chassis of existing cellular processes. So there are a number of questions that are relevant to this. What kinds of lifelike properties would be interesting to create in the laboratory? What this is sort of related to what is life? What do we mean by life? What interests us about it? What makes it different from other things? What features are needed to make a living system? So those properties, if we want to focus on making lifelike materials, what do we really want to pay attention to? Is it their self-replicated replication, or their metabolism, or their programmability, the fact that they're in vesicles? So I'd like you to be thinking also about what can be learned by exploring synthetic life, what kinds of applications can be enabled by creating these molecular cell-free systems. And some of the things that we'll be addressing are the sort of the state of the art, what kind of conceptual advances we need to learn how to create more complicated systems than the ones that we have right now, what kinds of design processes we use, and where the challenges are. So those are some of the overall questions that we'll be getting back to. We certainly won't answer them, but the format that I've chosen for today is to have 5 very quick presentations, 10 minutes each, that sort of span the range from people trying to sort of synthesize what might be prebiotic origin of life scenarios, making life from scratch, to people who are working on trying to program molecular self-assembly processes or digital circuits, and some of the processes in between. So the goal is for them to basically tell you sort of what is the state of the art in these cell-free systems, and if we have time, which I hope we'll have a few minutes at the end, we can get back to some of these overarching questions. So without further ado, I'll introduce the first speaker, Jason Shrum, who got his bachelor's in cellular and molecular biology at University of Michigan, also harpsichord, and is now a PhD candidate at Harvard with Jack Szostak. All right. So thank you, Eric. So I'm going to talk to you today about some of the things that I've been working on in Jack's lab. Some of this stuff was completed about a year ago, the data, so it's going to illustrate a lot of the conceptual principles on the protocell that we're working on in the lab, and then we'll move on to some more current things. So one of the questions is, what is a protocell? What are we thinking it is? What do we want from it, and all of that. So we have illustrated an image of a protocell. We're asking for a compartmentalized system here, so we want something where we can separate an external environment from an internal environment. This will spatially restrict, what's going on inside of it, the chemical evolution, which we would hope to transform into a biological evolution. We want our system to be capable of transforming chemical energy, so we can have some sort of a high energy form that's going to be driven, down a thermodynamic gradient to a more stable form. We want the potential to evolve a biological function, so some sort of structural role, maybe an aptamer or a catalytic role, something that's going to help drive, one of these protocells to compete with a neighboring protocell and emerge out of a population. We want it to replicate. We need heritable transmission from one generation to another. We want it to grow and to divide, finally. So this is the image now of our model protocell. How we have it illustrated here with an encapsulated primer template complex. It's encapsulated within a fatty acid vesicle.
The external environment, we have activated monomers. These are equilibrating across the vesicle membrane. Once they are inside of the vesicle, they're participating in the template copying reaction, which is shown in red as the single-stranded portion of our template is copied. The vesicle is growing as we have increase in osmotic pressure, which exerts a force on the internal wall of the vesicle. This vesicle acquires new fatty acids either from two means, non-competitive growth or competitive growth. Competitive is going to be taking fatty acids from vesicles that do not have productive reactions occurring in them. Non-competitive is going to be just free fatty acids in solution or micelles. Finally, once it reaches a certain size, we can have division into two daughter protocells, which completes the cycle. One of the questions is whether or not these fatty acid vesicles can actually retain oligonucleotides. Typically, they're thought of as being rather weak. They do hold them a 10-mer indefinitely. They're permeable to nucleotides. We show in blue just AMP, which is not permeable. As soon as you reduce the net charge with magnesium in AMP or ADP, which have different affinities for magnesium, we can see an increase in permeability over time to equilibration over the course of 24 hours. This is our replication scheme. So we have basically an RNA triphosphate, a standard RNA triphosphate here. We're looking at the 2' and 3' regions, as well as the 5' and manipulating those. So what we're choosing to do in this scenario is we're adding an amino group onto the 2' position. The 3' is deoxy. The 5' is activated with an imidazole. This acts as a better leaving group. The amino group is a better nucleophile. Once aligned on the template, these are spontaneously reacting in a chemical chain reaction to copy the template, leading to 2' 5' like phosphoramidate DNA. So this is a demonstration asking whether or not these analogs, does this actually work, or do we have to come up with an alternate chemistry? The assay that I'm asking for here is the addition of 4 analogs on a complementary template, and then I have each of the analogs illustrated either G, C, D, or U, and then I'm looking at 3 different template types, DNA, RNA, and LNA, and what we observe is the more A form the template is, LNA, all the way to B form, DNA, where the A form performs the best, B form is the weakest. This is a non-enzymatic optimization schematic, so we have a very wide pH range from 6.5 to 9. Temperature range from 0 to 35, a variety of templates. It's independent of, things that help enzymes work. A very clean reaction. So primer here, the fully copied template up at plus 15. So since our goal is to get this actually happening in vesicles, one thing, that the whole point is if we have a primer template complex encapsulated within a fatty acid vesicle, are we able to get these analogs permeating across the vesicle wall to participate in the reaction and complete a template copying reaction? So what I have illustrated here in the first panel is a reaction in solution. Time is shown in hours from 0 up to 24 hours. The second portion of this image shows a reaction happening in MAGMM, myristoylic acid, and then glycerol monoester myristoyliate vesicles. Panel B is a C10 system, a capric acid decanol monocaprin system. The significance of this is these are fatty acids that are actually recovered off of certain classes of meteorites. So you can imagine a scenario where you have a meteorite landing, some sort of prebiotic soup that's forming vesicles, possibly picking up nucleic acid polymers of some sort, and they're actually forming functional they're capable of forming functional protocells. C is another vesicle composition. This is a branched fatty acid alcohol, farnesol. And then D is a phospholipid vesicle, which is not compatible with this reaction. Phospholipids are, of course, formidable barriers to nucleotides and other sort of small solutes. So we did not expect that to be to allow template copying reactions to happen, and they do not. So the other thing that I'm talking about now is we want to know, we're going to have issues with the monomers and, fidelity and affinity for the template. So we want to look at, can we get productive things happening with oligonucleotides? It might be easier to incorporate mixed sequences, have a higher genetic variability, as well as copy longer templates. We can also take advantage of a number of inherent features of oligos, like the base stacking of the substrate, a higher affinity, lower concentrations, things like that. So I just have some quick examples that I'm showing here. A 3-mer, 4-mer, and 5-mer. where I'm asking for the addition of this, and we see what we would expect to happen with a 3-mer at a high concentration. The template copy reaction is completed in about 24 hours. As we increase the length of the oligo substrate, we can reduce the effective concentration as well as the time to completion. There are also, with these, we can take advantage of some small molecule reagents, like Nicholas Hudd worked with proflavin in a different non-enzymatic chemical ligation reaction. It was shown that was effective. There's also coralin, ethidium bromide, acridine orange is a few examples of a lot. I'm just showing here that proflavin is effective for the addition of a trinucleotide, a single addition reaction, where we actually double the efficiency of the ligation reaction compared to no proflavin. The vesicles are permeable to oligonucleotides. The trinucleotide is actually the cutoff. Tetranucleotides are not permeable. This graph is just illustrating permeability of a trinucleotide in the presence and absence of magnesium, where in the presence of magnesium, it's increased permeability versus without magnesium. The right panel is showing GMP in a different format. So this is my summary, which we have vesicles of mixed various amphiphil compositions that are able to retain nucleic acid polymers. They have selective permeability to small solutes. If we reduce the net charge, either with magnesium or in the case of our chemically activated substrates with imidazole on the 5' monophosphate, that increases the permeability. We can use these activated analogs to copy nucleic acid templates. The vesicle composition determines permissivity to the copying reaction, and we can get this to happen in prebiotically plausible amphiphiles. So that's all I'll talk about. I'd like to thank Jack in my lab. Sharif was a postdoc in our lab who's moved on now to the University of Denver, and at Harvard, Irene, and members of my committee, George, David, and Pam, and Eric for letting me talk.
Amazingly good timing. So I think we should permit one or two questions, quick questions, before we go on to the next speaker. Yes.
What's the mutation rate?
So we haven't actually addressed alpha-deli at this point. We've just been synthesizing the analogs. Those are actually time-consuming and rather challenging. So we actually just got the full set last week of all 4 of them. So I haven't actually done fidelity issues. I can say that if we try to do the reactions on a wrong template, nothing actually happens. So I think the fidelity is actually probably reasonable. I don't know exactly that.
Our next speaker is Mike Jewett. He got his PhD at Stanford from Jim Schwartz. I guess he did a postdoc briefly in Denmark.
That's right, for 2 years. Yeah.
And is now at Harvard with George Church. So please.
Excellent. So good afternoon. So I'm going to try, first of all, I'd like to go ahead and thank Eric again for inviting me to present. And I'm going to try to address and think about things on kind of the level that Eric was mentioning before in the context of a project that I'm working on with George Church at the Harvard Medical School. And that is we are working towards construction of a self-replicating entity from pure molecules. And how that is different from what Jason was describing just a minute ago is actually we are kind of starting with a protein world. So assuming that we already have, for example, a ribosome starting. So as you kind of take one step forward, that's where it's going. So one of the broader questions we need to begin to even think about in terms of in vitro synthetic biology is there even a need for it? Which is an interesting question, and we should be thinking about. And I, of course, am one of the parties that definitely feels there is a need. And I think that in vivo, what we find is that it's difficult to design, control, and insulate user-defined objectives from the host. We're getting much better at doing that, and by trying to use the tools that synthetic biologists are promoting, I think that we're moving in the right direction, where we are taking modern cells, learning how to reduce their complexity, and incorporate modules that allow us to take advantage of biological catalytic reactions. But one thing that should not be missed is that historically, decreasing the dependence on cells has led to increased engineering flexibility. So all of us, for example, are reliant on PCR, which is an in vitro reaction. So we don't necessarily have a PCR for proteins yet that works as efficiently for all conditions, but this is certainly a direction that one might be interested in moving towards. And building biological systems, if you kind of reverse the question, rather than taking a modern cell, can we build a biological system from the ground up, which would therefore reduce complexity and remove unnecessary overhead? So basically removing the things in the cell that make our jobs very difficult when we're trying to design things in vivo. And so the way we're approaching this and the overarching vision that we've taken is to move towards producing a synthetic entity that self-replicates from pure molecules. And so just as a little bit of a description about that, is what we'd like to do is have a minimal genome whereby we are defining this minimal genome through a biochemical approach and have arrived at a set of 150 genes. So that's kind of one of the questions Eric was asking, was what are the features that might be required for some of these minimal cell systems. For us, that number is about 150 genes from E. coli and its bacteriophages. And we believe that that would allow us to carry out both DNA synthesis, RNA synthesis, and protein synthesis. So when I say self-replicating entity, what we envision is a system that can carry out the activities of the central dogma. In a little more complex terms, it would look something like this, where you have many different reactions, all involved in protein synthesis, for example, including the ribosome, initiation factors, translation factors. And this work was originally described in a publication that came out in 2006 in Molecular Systems Biology. So what are the advantages of using and deliverables, for example, of constructing such a system? I think it's really important to kind of keep all of this in context as why are we doing this. From a cell-free point of view, and I think one of the biggest advantages of a cell-free system is that it allows you as an experimentalist to have direct access and control to the system that you're working with. So that can be easily modified, controlled, actively monitored, rapidly sampled. It doesn't have a membrane. So in fact, in the current movement of what we're trying to do, we're not focusing on a membrane. Ultimately, if you're trying to create minimal life, you would need that membrane to be there. But in terms of just having something self-replicate, we do not necessarily believe that that's necessary. One of the other advantages is from a bottom-up approach, you are defining all of the molecules in your system. So we actually know all of the 150 parts of our system. So allow for easier system-by-system debugging because all these parts are defined. What are some of the deliverables that we expect and hope for this system to afford us? First is test what we know and what we don't know. One of the greatest ways to truly know if you understand how the system works is to build it. So if you take a car engine, for example, out of a car, set it on the ground, take it apart into all of its pieces, put it back together, and it runs, you've proven to yourself that, in fact, I know what all the parts are doing and I know how this works. We are essentially suggesting the direction we're trying to take in vitro synthetic biology is along that route. That we'd like to take biology to its catalytic core, pull the pieces apart, learn how to put it back together, and get it running. Not to suggest that's easy, but that's kind of where we're going. One of the things and advantages of a platform like this is we believe it will generate a new way to look for antibiotics. So a lot of antibiotics towards bacterial systems, for example, are targeted towards the peptidyl-transferase center or also towards the tunnel where proteins come out. Here we'd be able to learn and understand how to target self-assembly of a ribosome. It also provides flexibility if you're trying to think about how you might be able to use an in vitro system for applications in producing biopolymers that are difficult to make in vivo because of either their toxicity, complexity, or unusual cofactor requirements. So basically, we're trying to take a stepwise modular approach to this where Brilliant. Thank you. Where we're starting with natural E. coli parts, we then would envision ourselves beginning to mix natural E. coli parts with in vitro made parts and ultimately simply in vitro made parts. Our top priority when you think about where do you start in such a system like this is we would like to construct a highly active synthetic ribosome. So what I mean by that is a ribosome that's been made by parts that have been entirely made in vitro. So one of the major challenges in this process, there are many, and I'm just going to highlight one as an example, is that in fact what we've shown and what's previously been shown and recorded in the literature is that reconstituted ribosomes, so if you take a native E. coli ribosome and you break it up into all of its parts, so a ribosome has 57 parts, bacterial ribosome. The small subunit has 21 proteins, 1 RNA molecule. The large subunit has 33 proteins and 2 RNA molecules. If you take an E. coli ribosome, break it into its parts, and put it back together, what's been shown in the literature is that is able to translate polyuridine into polyphenolalanine. So we don't even necessarily know if you take a native E. coli ribosome, break it into its parts, put it back together, if that can make a protein in an in vitro system. And in order to ultimately make protein in our self-replicating, or our synthetic replicon, we'll actually have to have a ribosome that can make protein in vitro. So one of the key first steps for us was to actually prove that if you take native parts, break it apart, put it back together, that in fact would allow you to make protein. And so just a brief touch on how that happens. So basically, functional ribosomes can be reconstituted from their native parts. This has been shown in the literature in the late 60s, where basically you can take a functional 30S subunit, you can break it up into its protein parts and its RNA parts. Through a one-step incubation process, you can reconstitute this into a functional 30S subunit. You can do the same thing with the larger subunit, which is the 50S, which has 2 ribosomal RNA parts and, in this case, 33 proteins. You can break it up into its individual component parts. You can then carry out a 2-step reconstitution reaction to get a synthetic, or to get a reconstituted ribosomal subunit. The real question for us was whether these reconstituted ribosomes could actually make protein. So what we went ahead and did is we put reconstituted ribosomes into a cell-free translation system, and what I'm just going to show you here are the results of that. So what we have shown in this case is that reconstituted 50S ribosomal subunits coupled with native 30S subunits express active luciferase. So what we've done is, on the y-axis, you'll see a model protein that we use just to report the production of protein. We have luciferase that we're using. The furthest bar is indicating a positive control where we have a native 50S subunit and a native 30S subunit that have come together, and we're producing approximately 450 nanograms per milliliter of protein. If you take a reconstituted 50S subunit, so one where you've taken all the ribosomal parts, you've reconstituted it into a functional unit, and you add that, couple it with a native 30S subunit, that produces protein. So in fact, it does not only produce polyphenolalanine, but in fact you can convince them to produce protein. We've added chaperones and see that that doesn't necessarily help, and these are simply 2 negative controls where you're only reconstituting either the RNA or the protein. We've gone on to show this both with the 30S as well as with the 70S subunits together. And in general, what we've been able to confirm is that reconstituted ribosomes can express full-length protein. What I haven't shown you is we've also been able to change the reconstitution protocols so that we can do this under physiological conditions, and are now beginning to focus on expanding our ability to build ribosomes using individual component parts. In the short term, just in terms of applications and where we think this is going, we believe that this work will add to the knowledge of describing ribosome activity and our understanding of how ribosomes work. There's also been a couple of very important papers here from Ueda's group demonstrating some of their results along these same types of lines, working with functional reconstitutions of 30S subunits. And looking forward, we believe that the synthetic replicon will catalyze a new paradigm for studying, tuning, and controlling life. So essentially gathering all of the catalytic core of biology. So just in terms of acknowledgments, I'd like to thank George, and my work has been funded by GTL and an NIH Pathway to Independence Award. So thank you.
Since we remain in pretty decent shape time-wise, let's take a question if there is one. So, yes, Christine.
I had just maybe 1 or 2 questions. You went through the last data slide pretty quickly. So you showed the one reconstituted subunit with a non-reconstituted subunit.
Right.
Have you tried both reconstituted subunits?
Right. So that's the
slide that you passed.
The next 2 slides that I passed. One is the complement, which would be a reconstituted 30S with a 50S, and then if you try them both together, and it still works.
So is it less? Do you see less translational activity with both reconstituted?
Yes, you do. So the number drops off by about 6-fold.
Okay. There's one behind.
So were you doing that with a single ribosome or a mixture? I'm just wondering if the activity at 50% is just because like half are not coming together, or are you just studying a single ribosome molecule?
Okay, so are these single molecule ribosome experiments, or do we have a pool of ribosomes that some are active and some are not?
Exactly.
We have a pool of ribosomes. Some are active, some are not. In fact, we're estimating at this point that about 1% of our reconstituted ribosomes, when we are carrying out a 70S reconstitution, so in which case we're mixing 57 parts together, we're estimating that we have about 0.5 to 1% of those actually functioning. And typically what if you base it on the amount of luciferases that we're producing, each ribosome of that 1% would be producing about 1 luciferase molecule. The trick is that in order to really know the answer to that question in terms of how many are active or not, you need to carry out polysome analysis, and that's something that we're looking forward to doing.
I have one question here, which is,
Okay.
what are the prospects for producing, basically transcribing and translating in vitro to generate the components of the ribosome rather than purifying them first and reconstituting?
Okay, so Eric kind of touches upon exactly where this project needs to be going, which is, okay, this is great if you're taking native parts and you're breaking them apart and you're sticking them back together. So the next thing we want to be doing is producing these parts in vitro and then having those come together. What we've shown historically, and I think that it's fair to say that in bacterial cell-free protein synthesis systems, whether they're crude extracts or in fact the pure system, that bacterial proteins actually express quite well. And when you're dealing with some of the most highly expressed proteins in the cell and that they're small, we don't think that we're going to have a tremendous amount of issue expressing them. The interesting element that comes to mind is how do you orchestrate the expression of these proteins with the ribosomal RNAs to have them self-assemble in the same mixture, which is quite a much larger question. Does that make sense?
Okay. Yeah. I was partly wondering if there are chemical modifications that this requires.
So in terms of the proteins, the chemical modifications are unimportant. Probably the major challenges with in vitro is with in vitro transcription of the 23S ribosomal RNA sequence. And that sequence actually requires 6 nucleoside modifications to be fully active in reconstitution reactions. So if we can move towards a more natural in situ production of a ribosome, that may be less important. But otherwise, we'll have to figure out how to evolve a 23S ribosomal RNA sequence away from that.
Thank you.